My name is Dale Kreitler, and I'm a scientist at the Automated Macromolecular Crystallography Beamline at the NSLS-II. And we help users or researchers from around the world determine the structure of biological macromolecules such as DNA, RNA, or protein. And we help researchers better understand the chemical systems that control life itself. So today I'm going to tell you a little bit about a project that we worked on with a group at Mount Sinai down the road in New York City, and what we did to help discover a new mechanism for combating pathogenic bacteria. So to give you a little background on the motivation for why we were interested in this project, antimicrobial resistance is an increasing problem. It's predominantly an issue in hospital-acquired infections, so perhaps you've had a friend or a relative that was impacted by a MRSA or C. diff infection while in the hospital. And the CDC estimates that it affects upwards of 3 million Americans per year. So there's an urgent need for chemical tools and strategies to combat this increasing resistance to conventional antibiotics. So one potential strategy that we could use to combat this increasing antimicrobial resistance is we could hijack the native bacterial defense mechanism for antiphage signaling. So what this is actually like a miniature immune system that bacteria have developed to protect themselves from phage viruses, and phages are viruses that infect bacteria. So what we could do here is actually activate a self-destruction sequence in the bacteria so that it would think that it was being attacked by a phage and it would initiate a self-destruction sequence. So in order to do that, we need to understand how that system works. So understanding this phage defense mechanism involves two components. So the initial step in this process is the bacteria has to detect foreign DNA, and it does this with an enzyme signaling pathway that creates a messenger signal. And this messenger signal then activates another enzyme which goes and chops up the foreign DNA. But we don't know what this messenger signal is, and we don't know how this messenger signal activates this complex. So that's what we set out to understand and to figure out. So there were two questions that we needed to address. First is which molecule activates this bacterial immune response. And so our collaborators at Mount Sinai figured out a way to analyze a bunch of different potential candidate molecules for activating this antiphage signaling system. And they ultimately found this cyclic AG molecule did the trick. So that's where we come in. So the Cap5 is this large protein complex that's involved in this antiphage signaling. So how does this molecule, this AG molecule, interact with the complex? And that's where we step in at AMX and NSLS-II. So we use X-rays, which have a very small wavelength, and they can be used to study proteins and things like that, which have atoms that are, very small distance apart. So to give you some background in the experiment that we run at AMX, first we have to generate crystals of these biological molecules. So in the upper left here, you'll see a droplet which has protein crystals in it. So crystals are repeating 3D patterns of molecules, and at AMX, we have a highly automated experimental end station, so we use a 6-axis robot arm and automation to scan these crystals in the X-ray beam. And based on how they diffract X-rays, we can determine the 3D structure of the molecules in the crystal lattice. So we actually collect these movies that are just a few seconds long, and then by analyzing the movies, we can determine the structure, and then ultimately the results are uploaded into public repositories like the PDB. So back to Cap5. So the molecular mechanism behind this bacterial self-defense mechanism, we discovered that the protein complex actually formed a tetramer when it interacted with this AG molecule. So AG, it binds to the protein, and then it assembles into this large aggregate, which then can carry out the nucleic acid hydrolysis. So you could think of it as a giant pair of molecular scissors, actually. So, and then on the right is the electron density that controls, or is ultimately what the users are focused on when they're doing the experiment. And these interactions that we observe in the density are what control this dimerization or association property of the protein complex. So once this molecule binds the protein, it chops up the DNA, and then the cell self-destructs, so to speak. And then ultimately, we wanted to test and evaluate whether we had the right molecule. So this Pseudomonas syringae was a model plant pathogen species. So on the top row is a normal culture of this bacteria, which has this antiphage signaling system. So in the absence of the signaling molecule, the bacteria grows, it's happy, it's doing its thing. And then on the bottom row, when we add this signaling molecule, the self-destruction pathway initiates, and then the bacteria doesn't grow anymore. So that's what you observe in the bottom row. So the system behaves how we would expect if we did in fact discover the correct signaling molecule. So conclusions, we found the signaling molecule Ag that initiates the self-destruction pathway, and using X-ray crystallography at AMX and NSLS-II, we were able to determine the structure of the complex that performs the nucleic acid hydrolysis. And then this is really important because this could be the basis for future antibiotic adjuvant therapeutics. You know, you would take this potential drug-like molecule, which would have a similar function to the signaling molecule, and it could help antibiotics more effectively kill these resistant bacteria. So I'd like to thank the CBMS and our support staff at NSLS-II, as well as our collaborators at Mount Sinai and our funding agencies.